Here are two high-yield USMLE-style questions focused on gene expression modifications in the context of transgenic strategies in mice, including random insertion and targeted gene modification. Question 1. A researcher is studying gene expression modifications in mice by creating a transgenic mouse model. The researcher uses homologous recombination to target a specific gene for insertion or deletion in the mouse genome. Which of the following best describes the mechanism of homologous recombination used in this experiment? A. The foreign gene is inserted randomly into the genome, potentially disrupting the function of other genes. B. The foreign gene is inserted at a specific locus in the genome through crossover events between the mouse gene and the introduced gene. C. The entire mouse genome is sequenced, and the foreign gene is inserted at the most accessible location. D. The foreign gene is transfected into the mouse egg and expressed as an episome outside the genome. E. A viral vector is used to integrate the foreign gene into the mouse genome via reverse transcription. Answer: B. The foreign gene is inserted at a specific locus in the genome through crossover events between the mouse gene and the introduced gene. Explanation: homologous recombination involves targeting a specific gene locus in the mouse genome by introducing a foreign gene that shares homology with the gene of interest. This allows the gene to be inserted or deleted at the specific location through a crossover event during cell division. This is a precise method in contrast to random insertion. Question 2. A transgenic mouse model is created by inserting a gene randomly into the mouse genome. This random insertion can potentially disrupt the function of genes near the insertion site. Which of the following is a disadvantage of this random gene insertion approach? A. The insertion is always precise and does not disrupt the surrounding genes. B. The inserted gene will always be expressed in all tissues of the mouse. C. The insertion site is unpredictable, which can result in unintended mutations or disruption of neighboring genes. D. The foreign gene may be deleted through natural selection during the mouse's embryonic development. E. Random insertion does not require the use of genetic modification techniques, such as CRISPR. Answer: C. The insertion site is unpredictable, which can result in unintended mutations or disruption of neighboring genes. Explanation: random insertion of a gene into the mouse genome can be problematic because the insertion site is not controlled, which may result in unintended mutations or disruption of neighboring genes. This lack of control can make it difficult to predict the precise effects of the inserted gene on the mouse's phenotype. Unlike homologous recombination, random insertion does not guarantee that the gene will be inserted at a specific, targeted location. These questions